Hi friends, today's topic is Krebs cycle. Krebs cycle, also known as citric acid cycle. In this cycle, the first product is citric acid, so it is called citric acid cycle. And citric acid cycle contains 3 carboxyl groups, and that is why it is also known as TCA cycle or tricarboxylic cycle. So Krebs cycle has several stages. In this video, I discuss about all stages. Okay. So at first, step 1. In case of step 1, acetyl coenzyme and oxaloacetic acid both are combined and to form citric acid. Acetyl coenzyme is 2 carbon compound, oxaloacetic acid is 4 carbon compound, and citric acid is 6 carbon compound. Acetyl coenzyme combines with oxaloacetic acid to form citric acid by using the enzyme citrate synthase. Okay, this is the simple stage. Next, step 2. In step 2, citric acid transferred into cis-aconitic acid by removing a water molecule, and cis-aconic acid transferred into isocitric acid by adding a water molecule. And this process is catalyzed by a single enzyme, just a single enzyme, which is called aconitase. Actually, these steps involves a hydration of citric acid to give rise to cis-aconic acid, and in this stage, cis-aconic acid further give rise to isocitric acid. Actually, the formation of cis-aconic acid from citric acid due to a asymmetric dehydration, and similarly the formation of isocitric acid from cis-aconic acid due to the result of stereospecific hydration. This is also a hydration. So this process is performed by a hydration. First one is asymmetric dehydration and the second one is stereospecific hydration. Okay. Citric acid to cis-aconic acid performed by asymmetric dehydration. Asymmetric. So I asymmetric dehydration, and isocitric acid forms formed from cis-aconic acid by stereospecific hydration. Okay. So next step 3, where isocitric acid is transformed into oxalosuccinic acid by the enzyme isocitric dehydrogenase and NADP+ or NAD+. It is the first oxidation step in Krebs cycle, where 2 electrons and 2 hydrogen ions removed from this substrate, isocitric acid, and this ultimately taken up by coenzyme NADP+ or NAD+ and formed NADPH plus H+ or NADH plus H+. Further, oxalosuccinic acid transferred into alpha ketogluteric acid by decarboxylation process. And alpha ketogluteric acid is most important acid for plant. Magnesium or manganese is required for decarboxylation reaction. All this actually step 1, step 2, step 3, all the reaction are reversible. So next is step 4, where alpha ketogluteric acid is transferred into succinic acid by oxidative decarboxylation process, where thiamine pyrophosphate or TPP is involved in the decarboxylation of alpha ketogluteric acid, and this TPP complex then reacts with lipoic acid to form succinic semialdehyde, and succinic semialdehyde then reacts with coenzyme to form succinyl coenzyme, and reduced lipoic acid. Then reduced lipoic acid is reoxidized by NAD+. Okay, and NAD+ containing enzymes, where NAD+ is reduced to NADH and H+ and CO2. Okay, and this process is irreversible, and the whole enzyme complex catalyzing these reactions is termed as alpha ketogluteric dehydrogenase. So the last stage of this reaction is the conversion of succinyl coenzyme to succinic acid, and it is also a second oxidation step, where the energy locked in succinyl coenzyme is released and utilized in the formation of an energy-rich pyrophosphate bond. Thus, in this conversion, there is a side chain where guanosine diphosphate (GDP) and inorganic phosphate GDP and inorganic phosphate unites to yield a molecule of guanosine triphosphate or GTP, and this reaction is reversible and controlled by enzyme which is succinyl thiokinase. Next, step 5, where succinic acid is converted into fumaric acid by the release of 2 electrons and 2 hydrogen ions, and it is the only reaction in Krebs cycle where 2 hydrogen ions are taken by ferriflavoprotein and not by any pyridine nucleotide, and FADH2 is converted in FAD is converted into FADH2 after taking 2 hydrogen ions, and succinic dehydrogenase helped in this reaction, and it is the third oxidation step in Krebs cycle. And next is the formation of malic acid from fumaric acid, and it occurs as a result of the addition of water molecule at the double bond of fumaric acid, and it is catalyzed by the enzyme fumarase. Okay, so next, step 7, it is the last step, and it is also a oxidation process where malic acid is oxidized to oxaloacetic acid by the enzyme malic dehydrogenase. Here NAD+ is reduced to NADH plus H+. This reaction completes the cycle, forming oxaloacetic acid, which then may continue in the Krebs cycle by reacting with acetyl coenzyme to form citric acid, and so on.